Good morning. For those who don't know me, my name is Filipa Ferraz. I'm the head of sector leading the work on ethics within ERC and have the pleasure to welcome you to our second online ERC Ethics Forum on behalf of our scientific director, José Labastida, who cannot be with us today. A few words about our seminar. The aim of this internal forum is to provide a platform for discussion, to help us stay abreast of ongoing discussions in research ethics, and to inform our work in ethics in order to best support our ERC grantees. Today's talk will be about genome editing, achievements, hopes, and limitations. Let me introduce today's speaker, Lluis Montoliu. He's graduated in biological sciences in '86 and obtained his PhD in molecular genetics in '90 at the University of Barcelona. Currently, he is a research scientist of the Spanish National Research Center, CSIC, and established his laboratory at the National Center for Biotechnology, so CNB, in Madrid since '97. After two postdoctoral periods in Heidelberg and Barcelona, in German Cancer Research Center and in Autonomous University of Barcelona, he continued working in this domain. Since 2007, he was also appointed researcher at the Spanish Research Initiative on Rare Diseases, where he is now serving at its steering committee. He has been honorary professor at the Autonomous University of Madrid for 20 years, exactly till 2 years ago. And since 2007, he is the director of the Spanish node of the European Mouse Mutant Archive. Including in his PhD, when he worked in plant molecular genetics, he has always been working on the genetic modification of organisms. Since '91, he has been working in several scientific projects within the field of animal transgenesis. At the CNB, he leads a research team interested in basic research to understand the mechanism controlling gene expression and organization in mammalian genomes, and in applied science, generating animal models for the study of human rare diseases, such as albinism. He has contributed significantly to animal transgenesis methods, developing artificial chromosome transgenesis. He has also pioneered the use and dissemination of gene editing CRISPR approaches in Spain for the functional analysis in vivo of regulatory elements found in the non-coding genome. Currently, he is involved in collaborative efforts towards the universal genetic diagnosis of all known forms of albinism and collaborates with patients' associations such as ALBA, the Spanish Association in support of people with albinism. He has published more than 110 articles, several books on genome editing and albinism, and co-invented several patents in the field. He is currently president of the European Society for Pigment Cell Research, president of the Association for Responsible Research and Innovation in Genome Editing, ARRIGE, and serves at the board of some additional societies. In 2006, he founded the International Society for Transgenic Technologies, for which he has served as president since its inception in 2014. He is a long-standing member of the CSIC Ethics Committee, and since July this year, he was appointed president of the CSIC Ethics Committee. I also have the pleasure to highlight that over several years, he served as experts in our ERC ethics panels. In addition to research, he is also interested in biotechnics, science education, and popular science, and received several prestigious awards in this area, So many that I will not name each one of them. If not, we would not have conference today. So before we start, let me tell you that we will have a question and answer after the talk. Please ask your questions directly in the chat and with your name if you would like. The recording of our seminar will stay available on this YouTube channel afterwards. Lluis, we are really honored to have you with us today. So the floor is yours.
Many thanks for this very kind introduction, Philippa, and many thanks to the ERC Ethics Sector and to Nicole and to everyone for this invitation. So without further delay, let's start with the seminar. So I will start sharing my screen, and then you should see the first slide of my seminar. So today, we're here to discuss about genome editing, achievements, hopes, and limitations. And the first thing I think I need to say is that we need to share and we need to congratulate these two researchers. This is the first time two women researchers were awarded the Nobel Prize. On the left, Emmanuelle Charpentier, the French researcher now working in Berlin. On the right, Jennifer Doudna, the American researcher worked at the University California, Berkeley. They were, as you know, they were awarded at the beginning of October the Nobel Prize in Chemistry this year for the development of a method for genome editing. So CRISPR, so this fantastic, the magic word, was not mentioned by the Swedish Academy, but CRISPR was just behind. They said a method because they meant that there is not a single method. There's several methods for conducting this genome editing. I will only be referring to the most amenable, the most affordable, and the most efficient method, which we know this is CRISPR-Cas. But before CRISPR-Cas, we have three other methods, such as the meganuclease, the zinc finger nucleases, or the tail nucleases. So CRISPR, we need to just say a few words that they're coming from the prokaryotic world. They're coming from bacteria. And they were discovered in Spain, in the south of Spain, by this microbiologist, Francisco Juan Martínez Mojica, and he's a microbiologist at the University of Alicante. He was the first to realize that there was some peculiar organization in the genome of these bacteria in which they were holding pieces of viral genomes. And when this was happening, the bacteria were resistant to the same viruses. So in the same manner, he was discovering an immune system, a defense system that had a genetic basis in the bacteria. He was also naming this process with the CRISPR, and this is this word that has been successful and is in the mouth of everyone. So for 20 years, between the discovery of the CRISPR systems made in 1993 and the demonstration that this could be used as a genome editing tool, at least these 12 researchers were involved in the progress. Of course, at the central, we have Emmanuelle Charpentier and Jennifer Doudna, but also we need to realize that there was a lot of basic science before, a lot of biochemists and molecular biologists and microbiologists that were contributing, so that in the summer of 2012, when these two researchers published their seminal paper, they could really make this proposal. I want to highlight the four geneticists that are in the last row: Feng Zhang, George Church, Rudy Jenisch, and Qi Yang. They were the first demonstrating that the proposal made by the two researchers was a reality. So starting in January 2013, so a bit more than 7 years ago, Feng Zhang and George Church confirmed that this defense system in bacteria could be used as a genome editing tool. So in bacteria, as we said, this is acting as a defense system. This has been going on for billions of years. And recently, we just realized that we can take this out of context and we can convert it into an efficient genome editing tool. This is how we learn about this tool in summer 2012, thanks to Charpentier and Doudna. It is a very simple, and if you allow me to say, beautiful system. It is only a binary system. It has this yellow patch. This is a nuclease, normally Cas9. Nuclease, it means it cuts DNA, but it doesn't cut randomly. It cuts where it is dictated, it is guided by this blue molecule. It's a small RNA that pairs with your favorite gene. So it's cutting that particular gene. So 8 years later, things have been evolving a bit. And now we're using, rather than 2, we're using 3 elements. The guide RNA, we have split it in 2 different molecules. And the most important thing to say here is that these 3 molecules, the nuclease, the CRISPR RNA, and the tracer RNA, which by the way was discovered by Emmanuelle Charpentier, everything is commercial. It means it's very easy to everyone around the world to purchase these elements and to conduct the experiment. So what is the action? What is the function of these CRISPR-Cas molecules? Well, they cut DNA. If they cut DNA, they are promoting the gene replacement in a manner that it was unforeseen before. This is a very old slide that I've been using for more than 25 years. This is why it looks a bit vintage. But this is what we have been always looking at the genetic modification. When we had a non-functional gene, we wanted to replace this non-functional by the corresponding functional copy instead of randomly inserting the copy. And this was a problem because normally without nucleases on the right, without nucleases, this was only happening 1 every million times. So this is a very, very low probability. But look what happens on your right, on your left, look what happens when you start using things like the CRISPR or any other nucleases. As soon as you cut a given gene, you increase this probability 1,000 to 100,000 times. So now the probability is 1 in 10, 10 to the minus 1. So this is really illustrating why we just ran crazy with this system. And this is why every single lab in the world is now using this system. So we're cutting the DNA, and what happens later on, the cell has to fix this aggregation, this insult. We have 2 ways of correcting or fixing this double strand break. So on the left, we can use the so-called non-homologous end joining process, which proceeds randomly, adding and removing letters, adding and removing nucleotides, indels, insertion and deletions. And eventually, there is some microhomology found, A in front of a T, G in front of a C, and so the chromosome is sealed again. But because we have added and removed letters, normally the gene is no longer working. But now it becomes the easiest way of inactivating a gene. On the right, this is what we all wanted to occur in most of the times, but unfortunately is not working most of the times. This is the homology-driven repair, in which we can provide a sequence, we can provide an oligonucleotide, sharing homology at either side of the double strand break, and adding the novo sequence, this short blue piece, which wasn't there before, and this we can use to introduce a mutation, or better, to correct this mutation. That would be a gene addition, that would be an innovative gene therapy. So there's a fantastic amount of applications that we can do with genome editing, particularly with CRISPR, deleting, inserting, replacement in biology and biomedicine and biotechnology. Let's go one by one very briefly so that we understand what we're dealing here. Disrupting a gene. So this is a trivial experiment nowadays. I never thought I would say something like this, but this is a trivial experiment. You just need to plan a CRISPR-Cas with a corresponding guide RNA to your favorite gene, and the gene becomes inactivated. Well, you can do this one by one, or you can do in thousands. So this is what they've done in Australia with this yellowfish, in which they target almost all genes that we have in our genome in order to discover an antidote for the poison that is associated with this yellowfish. They give the poison to cells in which they have inactivated each cell one gene, one of our more than 80,000 genes, and most of the cells died because of the poison, but only those that survived were those that were just leading to the description of new antidotes. If we combine more than one CRISPR-Cas system, we can promote deletions. And this is very interesting because this is, for instance, what it was proposed beginning of 2016 in animal models, in mice, for a very severe rare disease. This is this Duchenne muscular dystrophy, in which there is an animal model, there is a mouse model, and they target the dystrophin gene, and they remove only the exon that was carrying the mutation, leaving behind a shorter protein, but still functional. So this is something that we can do now with CRISPR. And also we can address our analysis in the non-coding genome, which is, as you know, the majority of our genome. 98% of our genome and of the mouse genome is non-coding, but it's full of regulatory elements. We didn't have tools for investigate these elements. Now we can target with 2 CRISPR elements at either side, and we can promote the deletion. So this is, for instance, what we do in our lab. We're working in pigmentation genes. The mouse line behind, it's fully pigmented, and the one on the top is gray. It's not gray because we have mutated the gene. It's gray because we have mutated a regulatory element, thereby demonstrating the relevant role of these elements. We can do inversions, so that was very difficult to do before. We can do any other chromosome rearrangement, such duplications on all the different rests. We can do very subtle things, and this is something I want to just stress, because this is really changed the life of many biomedical laboratories. So introducing fine mutations. So this is what we call the avatar CRISPR mice. And I use the metaphor of this science fiction movie from James Cameron. Remember these blue bees that were connected, each one with a human being? And I'm using this metaphor because now I can diagnose a mutation in a patient, and I can transfer exactly the same mutation to the corresponding mouse. So this mouse becomes the avatar of that person. We've done this many times. This is one of our patients in albinism, this rare disease that we are investigating. And in this case, the gene affected is just indicated. And what happens to this girl is that she's missing just a single letter. And this letter, exactly the same letter, has been removed to the mouse shown above. So now we can validate treatments. We can see whether small molecules or whether pre-existing drugs that can be repositioned can be a valid treatment for this without running the risk to exposing these patients to the drugs. We can use these avatar mice. We can do knock-ins. We can introduce big pieces of DNA, and we can inactivate the nuclease. So what happens if we are enabling the scissors to cut? So what's the point of having a scissors that do not cut? Well, the scissors might not cut, but may still sit somewhere in the genome. So we can combine this no longer cutting Cas9, so-called that Cas9, with activating domains, so we can promote epigenetic modifications of a gene. We can activate, and of course we can repress genes. So this is already opening a new era of investigations. We can also combine this dead Cas9 that is no longer cutting with other chemical activities. And this is something very fascinating that has been proposed from Boston by David Liu from the Broad Institute, such as using the deaminases. The deaminases, they're allowing to change directly some letters from C to a T, or an A to a G, without cutting the DNA. This has been progressing, and as you know, we can have in this experiment, we can change the letters of a given gene. We still don't know how to precisely identify the letter we want to change. There will be some changes in the letter which are sitting nearby, but I'm sure with a matter of time, this will be improved. We can even program these tools, these genome editing tools, to work in mitochondria. You know that mitochondria, they are derived from ancient prokaryotic organisms that were grouped together in a eukaryotic cell. And the thing you need to know is that CRISPR, in principle, is not working inside mitochondria. But David Liu just found a way, and this is just very recent, this summer, in which he went back using the tail system, and he was combining the tail system with this deaminase, so that now we can edit. It's not CRISPR, but is analogous. It's a combination of a tail with a deaminase, and you can now edit the mitochondrial DNA, which is important for a number of diseases that are associated with mitochondria. We have even more fancy evolutions of these variants of CRISPR, such as combining with a reverse transcriptase. And this is the so-called prime editing. We learned also from David Liu. Is using a very long guide that is pairing on a given gene, but also the other end is pairing with the opposite DNA strand, and it's leading the synthesis of new DNA because it's composed of a reverse transcriptase. So at the moment, we have a variety of different tools that are available to us, and we can use them for many different applications. I will highlight some applications later on in my talk, but here it's sufficient to say that we can use it for biology, for biomedicine, and for biotechnology. Well, we need to know not only what we can do, but we need to know the limitations of these tools. And this is very important because we need to be aware what we still cannot control, and we need to be aware about the different outputs that we will be generating from these experiments. The first thing to take into account is that we might be targeting similar genes. The guide RNA that we're using is using homology, and it's pairing with our favorite gene. But of course, the guide can pair maybe with one letter less, or maybe a couple of letters less somewhere else in the genome. And this is another gene that we didn't want to interrupt, we didn't want to interfere with, but we ended up editing. So this is the so-called off-target effects, which, by the way, are not that relevant because as we progress, there have been more than 100 programs available that are now ready for designing our guides RNA. So as we advance, there are better programs and we are selecting better guides. So sooner or later, I think the off targets will be fixed and we can deal. The other problem, which it's very difficult to fix, it's not the off targets, but the on targets. And this is associated with mosaicism. I told you before that when you are doing this double strand break, you have to fix this break. And there were two pathways, two main pathways. On the left, this non-homologous enjoining, and on the right, the homology-driven. We wish all the fixations and all the repairing would be occurring following this right path. But this is not the preferred repairing pathway, unfortunately. And this is only happening in very few cells, in particular those that are dividing. For the rest of the cells, we are left with this non-homologous, which is random, which means that every time the system has to fix this double strand, it will introduce some noise. It will introduce some variations. So this is why we have this mosaicism. So the best way to see what is the normal output of a genome editing experiment, it is just to show what's going on. This is sequence analysis. Every line that you see, it's a different mouse. This is an experiment we did in our lab. And as you can see, it is the same experiment, but every mouse is carrying a different mutation. And among which you have, of course, those that you are interested, but you need to scan through and you need to select. In other words, I'm using a pigmentation gene. I'm restoring the alveolus. And I hope this mouse will be uniformly pigmented, but it is not, it's patchy. And why is patchy? Because not in all cells, the correct function has been restored. I was injecting these CRISPR-Cas tools when the embryo was a one-cell embryo, but these CRISPR tools did not fade away, did not vanish. They remain as the embryo was dividing. So we have a morula with 8 cells, and each cell has, as we know, 2 alleles. So 8 times 2 makes 16 different ways the gene can be corrected, and then the resulting cells, they will be show up in the adult, and we will have these mosaicism. Of course, this mosaicism is not a problem at all in the lab, in the academic world, because we are producing mosaic-founder mice, and we are sequencing them, and then we're locating the one that carries our mutation of interest, and we can set the breeding, and then the next generation, we hope that the different alleles will be segregated until we can select the one and establish the colony. So the only thing to take into account here from the ethics point of view is that we will be probably generating more animals. We will be using more animals than we used to do before. But this is something that we can handle. This is something we can do. This is not something we can transfer directly to human beings. We cannot pretend to treat a person and say, we are not sure whether you will be benefiting, but for sure, maybe one of your children will. Well, this is not working like this in the hospital. It's not scientifically prudent. It's not ethically acceptable. And so this is the main limitation we need to be aware. This is what makes a whole of a difference between the academy and the reality in the clinic world. Of course, this is not only happening with mice, but this is also happening with plants. With plants, we are also producing mosaic plants, but very easily we can segregate the alleles that we want, and we can remove the rest of alleles that we are not interested in until we select the plant that is being genome edited according to our will. Sometimes this variety has some positive effects. This is a very specific example of the contrary. This is a rare disease. It's called Fanconi anemia, and it's a severe disease that is compromising life of the children affected, but is associated with the stem cells in the hematopoietic system. Of course, when you are using CRISPR to reconstitute the function of these proteins, because they have a role in the survival of the cells, even though you will have a mosaic distribution, even though you will have all the genetic variability, those that are correctly restored, they will have a survival advantage. They will have a proliferation advantage. And in this case, this variety will be of use and will be of help. And eventually this can be used for treating these children. So let's talk about different applications. And of course, the first one that comes to our mind is using these genome editing tools for gene therapy. So we know where the mutation is. We use CRISPR to promote a cat very near this mutation, and we're using our donor DNA, a template that carries the correct sequence, and we hope that everything goes fine, and then eventually the correct sequence is being included. Well, the first thing to say about gene therapy is that we have legal and illegal ways of doing gene therapy, at least in Spain, and I'm sure in other European countries. As we know, we are in Spain, we're signing this Asturias Convention in 1997 that is prohibiting the modification of the genome of our descendants. So we cannot modify the genome of the germline, but we can, of course, modify the somatic cells, either in vivo or ex vivo. And this is something to take into account when we always shocked in November 2018, when this person, He Jiankui, this researcher from Shenzhen in China, he was applying these CRISPR tools for genome editing human embryos. By the way, this is something that can be done in most European countries, including ours. We have laws that permit this, but provided that these embryos are not implanted in the women. So you have to destroy these embryos at the end of the experiment. So in the opposite, this He Jiankui, after editing these embryos, he was implanting, and as we know now, this was an irresponsible experiment that should not have happened, that led to the birth of 3 girls. There was 3 girls that we know. He was intending to block the CCR5 gene that encodes a receptor that is being used by the human immunodeficiency virus causing AIDS, so that he will be generating children that would be resistant to AIDS. Now, I want you to picture the mouse I show you with patching, so with patches black and white, and now you have to realize that these girls were also mosaic. It means that some of the cells had the CCR5 gene interrupted, but some of the cells had the CCR5 gene okay, and some of the cells had the CCR5 with other mutations. So the same variations that we have. But this is something we can handle with mouse models or with plants, but this is totally irresponsible. So this risk, this uncertainty, it is absolutely irresponsible. That's why he ended up in jail, paying a good fine, and what is most importantly, inhabilitated for life. Now we know, confirmed in several studies that were made available this summer, that all what we observe in mouse embryos and in other species can also be replicated in human embryos. Why we would be different? We are not different. So if you apply genome editing to human embryos, you will end up with off-target effects. You will be creating new mutations. You will have mosaicism. You will have chromosomal rearrangements, which means it's not only illegal, but unnecessary, but unwise to apply at the moment this CRISPR for therapeutic use in human embryos. And I'm saying unnecessary because there are other techniques, such as pre-implantation genetic diagnostic tests that we can apply very easily and very robustly. This has been already dealt with in numerous reports on the human genome editing, most notably in 2017, the first report from the National Academy of Science. Here in Europe, we have created this society. This is ARISH, the Association for Responsible Research and Innovation in Genome Editing, that I invite you to visit, where we discuss about the responsible use of these genome editing tools. And most recently, in September, the National Academy of Science, together with the National Academy of Medicine and the Royal Society, they put forward this updated heritable human genome editing report, defining clearly where you can apply these methods and what would be the limitations and the conditions. As it stands now in the world, so this has been very recently published by Baylis et al. So there's very few countries where you can even think about doing human genome editing at the embryonic level. Most of the countries are red, which means they are prohibiting. Some countries are yellow, which means there are some exceptions, and that applies to Belgium and Italy and Colombia, for instance. And the most disturbing things are the gray countries, where we don't know what's going on, where we don't know what is being applied. On the contrary, using these human embryos, we have thousands of them in many countries for research purposes, provided that we don't transplant them, we don't implant them in women, we use them for research. It is legal. It is legal in most countries. It's necessary. And why? So this has been proven as something very beneficial, such as this seminal work published in 2017 by Katiniakan. And Katiniakan just demonstrated that we need to do the experiments if we want to understand the role of some genes in the pre-implantation process, which we don't understand at all in the human embryos. And we take for granted that what we know from mouse embryos will be also the same with the human embryos. And this paper clearly demonstrated was not the case. So let me tell you a bit this traffic lights slide, because it will tell you that not only that we need to be double checking and double thinking before applying these techniques everywhere. So, but we need to be aware about the things that we can do and the things that we should not be doing. So we have a green light for applying, and this is where most of us researchers that we will be agreeing. We have a green light where we can apply these techniques for editing, for curing, or for preventing a disease at the somatic level. That means treating a given patient. We have a yellow light, which means it's mostly prohibited in many countries, but it might be allowed in some countries as soon as we advance in the scientific terms, which will be doing the same, curing or preventing a disease, but at the germline level. Not possible at the moment, not recommendable at the moment, but maybe in the near future. And certainly we have, and I think that most of us, we would agree on the red row. The red row, it means that we should not be applying these techniques for somatic cell editing or for germline editing, not for therapeutic uses, but for enhancement. And enhancement, it means this is an euphemism of the eugenetics. This is something we need to be aware, because there is a new philosophical movement, well, rather new, it's called transhumanism. And as you know, they are proposing that there should be They should be allowed to use all the benefits of the technique in order to become superwomen and supermen, in order to get additional capacities. And they are also triggered by some researchers that I admire, but sometimes they have these oddities. For instance, George Church is publishing a list of genes that he thinks that he will allow us to get additional capacities. So let's say you would be mutating the SCN9A, you will be insensible to pain. I don't think this is a super capacity. I think it is a problem. But one has to remember that we have only 20,000 genes. It means that most of the genes, they do many different functions. So this is called pleiotropy. So it's very naive to assume that inactivating a gene will have only one consequence. They might have many consequences. This is why it is dangerous. And we need to be aware of this because we have biohackers. We have biohackers such as this person, Josiah Sina. You can see him in this Netflix documentary on natural selection, which I recommend seeing, in which he says, and he pretends to be injecting this CRISPR-Cas in cells. He wants to inactivate the myostatin. It's a negative regulator of the muscle proliferation. And he wants to become something like this, the blue Belgium cow. And well, this is absolutely irresponsible and most dangerous, because besides this cow being very appreciated from the food perspective, I mean, the animal welfare associated with this animal is very, very complex and very difficult because, of course, not having this myostatin has consequences. This is something we need to take into account. So if we just leave apart the germline and we just focus on somatic, remember that we have in vivo or ex vivo treatments. Either we can deliver these tools into the patient, or we can extract cells from the patient, edit the cells outside the patient, and then return them to the patient. Well, believe it or not, today we have 43 clinical assays that they are already using CRISPR. They are clinical assays, they are not treatments. It means they are still being investigated. Most of them, they are ex vivo, a few of them, they are in vivo, such as this treatment or this potential treatment for Leber's congenital amaurosis, which is a retinal degeneration congenital disease in which they're injecting intraocularly and subretinally to fix the mutation associated with the gene, or with the immunotherapy. This is probably one of the future uses of CRISPR in the clinic. So this is immunotherapy. This is people with refractory cancers, myeloma, sarcoma, melanoma. You can get T cells, you can get lymphocytes from these patients. You can use CRISPR to inactivate genes that are negative regulators, so making these lymphocytes stronger, and you can add some additional receptors so that they can target more efficiently the cancer cells. So that's probably, and I predict this will be a good use of this CRISPR in the clinics. Or as we just learned 2 days ago, and this has been published in New England Journal of Medicine just on Saturday, and this is the first confirmation that this therapy works for sickle cell anemia and beta thalassemia patients. As you know, they have the beta globin of the hemoglobin, they have it affected, and the idea behind this CRISPR approach is to reactivate a fetal gene, the gene that is normally not active in the adulthood, is not active because it's repressed. But if we're using CRISPR for targeting the repressor, we're promoting what it was repressed. And this is exactly what they're doing. And now we have people that have been treated this way and then no longer require transfusions. In most of these cases, they're using these viral vectors, such as adeno-associated virus, to deliver these tools to the people. And there are some few exceptions, and this is also very recent for October. This is another company that is not only using, viral particles, but nanoparticles, nanotechnology. This is for treating this congenital transthyretin amyloidosis, which is another rare disease. We also need to be aware that we need to be very carefully select the nuclease that we're using. Matt Porteous was the first one to realize that even though we use in the lab the Cas9 from Streptococcus pyogenes or Staphylococcus aureus, these are pathogens for us. And it means that our immune system, they know these bacteria, they have antibodies. It was anticipating we would have antibodies against these nucleases, and we do. Most of us, we do, and we have T cells, so that would make it impossible to use these nucleases for treatment. So he was envisaging the use of some immunosuppressive drugs, or we can use CRISPR-Cas systems from many other bacteria. There are hundreds of thousands of species that yet to be identified, so that we can use in order to have new CRISPR-Cas, such as this is very recent, and this is coming from Jennifer Doudna. This is from this summer. She was describing some Cas proteins that were coming from huge phages that are infecting bacteria and are using the CRISPR system for fight other viruses that are infecting the same bacteria. And the thing is that these smaller Cas proteins, less than 1,000 amino acids, they can also be useful for gene therapy. So let's go beyond biomedicine, because I wanted to touch upon some other applications that are beyond the biomedicine, such as, for instance, so CRISPR can also be used for fighting vector, so that is insects-borne infectious diseases. We're talking about malaria, yellow fever, Zika, chikungunya, and dengue. And how do you do this? Well, basically, you are targeting some parts of the cell cycle of this mosquito. So if you are preventing the mosquito to reproduce, so the mosquito will disappear in a given environment and will no longer be transmitted. And this is done because if we compare what is done with the classical transgenic mosquitoes, according to Mendelian inheritance, they will rapidly be disseminated and but will rapidly will be diffused in the population. So it's very unsuccessful to distribute transgenic mosquitoes. Whereas if you're using CRISPR, the CRISPR, thanks to this gene drive effect, it will be converting all the alleles in the population, and very soon you will have all the mosquitoes that will be carrying these mutations. This is something that is being explored. It's not yet in the fields. Transgenic mosquitoes, they are already in the fields. These genome-edited mosquitoes, they're not in the fields, but I predict they will be soon there. What about animal biotechnology? Well, we could use this for preventing the infection of the respiratory and reproductive viruses that is affecting pigs. These viruses is using a given receptor. You can target this receptor with CRISPR, and then you run the resulting pigs that are no longer sensible to this pathogen. Or you can apply this in xenotransplantation. As you know, this is a very unseen proposal of using animals, organs from animals for our shortage of organs in human beings. The problem being that the cells from the pigs, they carry retroviruses, the PERP, and now George Church in 2017 was using CRISPR to inactivate the PERPs, and he was generating some pigs, as in the ones in the picture, that can now be used for with further genetic modifications for genomic transplantation. What about implants? What about the implant biotechnology? Well, this is a Spanish example. This is my colleague Francisco Barro from Córdoba in the south of Spain, and he's using CRISPR for targeting the gliadin genes in the wheat. And the gliadins are the ones that are forming the gluten. So if you're targeting the gliadin, you are producing a wheat that has a low content in gluten and can be eaten by people with celiac diseases. This is one of the things you can do with CRISPR. Or this is done in Japan, that is preventing premature germination. When you are about to harvest, let's say, this wheat, there is a sudden rainfall, and there is so much rain that the grains are beginning to germinate in the plant, so rendering the harvesting no longer possible. But if you know the genes that are associated with this premature germination, you can inactivate them and you can prevent this from happening. So this is a benefit for these people producing wheat in these regions. Or for instance, in the US, you can inactivate with CRISPR a given gene, it's called waxy, that is changing the composition of these corns, and it's producing almost 100% amylopectin, which is a beneficial product for food and for derivative industries. Or for instance, this is done in China, in which you can use CRISPR to delay the tomato ripening, which is also a threat that has some commercial consequences. So you are inactivated gene that are associated with ripening, and then for the same amount of days, the tomato is not ripening, and so you can distribute it to longer distances. Well, what happens in Europe is that, as you know, at the end of July 2018, we heard the sentence from the Court of Justice, and that basically what they said is that the genome-edited organism is similar to transgenic, and they have similar risk. So the full Directive 2001/18 applies to them. Well, actually, what they said, this is here, it's press, organisms obtained by mutagenesis are GMOs, and are in principle subject to the obligations laid down by the GMO directive. And they even added that they are not included in the exception that other mutagenesis techniques are normally used, such as the chemical mutagenesis or the physical radiation mutagenesis. Well, let me tell you that that was a legal decision that I don't have anything to say from the legal point of view, but is scientifically nonsense. And I want to be very clear about this, because we're not talking about transgenics when we're dealing with genome editing. Genome editing can also be used for doing transgenics, but in principle, what we do with genome editing, look at this. When we're adding a piece of DNA, we obtain a transgenic organism, and this is what we call a transgenic. When we are analyzing different variants, different varieties, different races, is because they have different letters in the same gene. And we approve them, and we don't have any problem. Of course, they are not transgenic. But now we can use CRISPR to change the letter so that we can change the characteristics of a race to become the desired characteristics of another race. Of course, this is not a transgenic organism, and this is now being regulated as such. Also, we need to be aware that with CRISPR, in one generation, we can transfer the desired character from one strain to another. Whereas without CRISPR, we have to go all the way through different rounds of backcrossing, and we will never be 100% sure. This is why many different scientific institutions have opposed to this decision, and they are trying to force to a reconsideration. I know that the European Commission is now investigating this issue, and it's planned that next April 2021, there will be a new document or a new decision. And before that, I hope we will also hear from the European groups of ethics that I know they are investigating. And finally, in this aspect, we need to remind that we're living in a global world. So what we do in our little corner of the world, what we do in Europe, doesn't apply to the rest of the world. We might be very strict on regulating these issues at the European level, but these techniques are already being applied in America, in Asia, in Australia, in Japan, in Korea. So we might be preventing our companies to grow these plants or to use these technologies in their animals, but we will end up purchasing all these different products from these different external companies. So let me finish for the rest of the minutes I have by telling you that CRISPR is so amenable, so there's so many applications associated to CRISPR that we can even think about CRISPR applications for the current pandemics, COVID-19, because there are some diagnostic applications associated with CRISPR. So what about CRISPR and COVID-19? All what I've told you so far refers to what we know as CRISPR 1.0. So we cut and we edit DNA. But you need to know that since 2017, and thanks to Feng Zhang, we can cut and edit RNA. So we are now having new nucleases that can edit RNA without touching at all the DNA. And this is something that was discovered just by chance by Feng Zhang. And he was using a new Cas protein called Cas13a, and it's cutting RNA guided by small RNA. When this guide RNA was finding the complementary sequence, the Cas13 become activated, was cutting the target RNA, but also become crazy. And this craziness, it was converted that this Cas13, once it was activated, was cutting everything else in the mixture. So this normally would have been a fiasco for most of our researchers, but Feng Zhang converted this fiasco into a diagnostic opportunity because he was spiking this mixture with some short RNAs that carrying a fluorescence marker, an inhibitor. And once they're cut, the fluorescent level is released. So you can combine now the detection of a given RNA with fluorescence. And so he was proposing the Sherlock. So Sherlock is a new diagnostic system that was followed the year later, 2018, by Jennifer Doudna with an equivalent system detector that was using the Cas12a. This is another nuclease. It's cutting DNA, but it's also turning crazy. And it's also cutting single-strand DNA, so it can be converted in the same way. It can also be converted in this chemistry, in this lateral flow. So this is like this pregnancy test that we are all used. We go to the pharmacy, we purchase this pregnancy test with a bit of urine, and we know whether we're pregnant or not. So we can apply the same techniques now with CRISPR for detecting. So as early as in March, Feng Zhang was already proposing to use Sherlock to detect COVID, different genes of the SARS-CoV-2 coronavirus with a very decent sensitivity, up to 10 copies per microliter. That was already superseded by this new method with a very interesting acronym, Carmen, that is combining Sherlock with microfluidics. These are nanodroplets. The only thing you need to know about this is that it allows you to just do a COVID-19 CRISPR detection 1,000 people at a time. So you can do 1,000 tests at a single time, so you can progress much faster with different colors. Regarding detector, you can also apply these methods. This is a similar way, instead in this case using Cas12. And if we are asking ourselves what is the difference and the comparisons between CRISPR detecting or RT-PCR detecting, this the gold standard for COVID-19, we need to know that these are faster, simpler, less sensitive. With PCR, you can go down to 1 copy of the virus per microliter. With CRISPR, you can go normally up to 10 copies. Normally, you are around 100 copies. So they are less sensitive and they are qualitative. They're not quantitative. RT-PCR is providing quantitative data, even though nobody is using this quantitative data, because the only thing we know when we do a RT-PCR is whether we are positive or negative. But there's always another information about the viral load that we are carrying. In May, the Feng Zhang was improving this system and was simplifying. So this system was called Stop COVID, and is as simple as you can see in the picture, because the only thing you need is a heater to maintain a given temperature for 45 or 60 minutes, some pipettes and some stripes for doing the procedure. So it's very easy. This can be conducted in many different places, not necessarily in a sophisticated hospital room. This system has been already approved for emergency use by the FDA, and unfortunately, the same has not happened by the EMA. So the European Medical Agency has not yet approved this. So now in America, they can already use these systems, but we cannot use. The last, and this is something that was published on Friday. So this is very, very recent. The last improvement in this system is coming from Jennifer Doudna. So the laureate researcher receiving the Nobel Prize was also using Feng Zhang's protein. So this is interesting. Jennifer is using Feng Zhang's protein, Cas13a, for this system in which you can combine a fluorescent detection of this virus with your mobile phone, with your smartphone. And so this is very interesting. So they are not amplifying. Basically, they are increasing the sensitivity of the method by not only using one guide, but using multiple guides. So the genome of the virus is cut at different places. And so there are many opportunities for this Cas13a activated to cut the spike probes. And these spike probes will end up with a fluorescence. And this fluorescence that you can detect with a given sensitivity, basically around 100 copies of the virus per microliter. And this is something you can do with your smartphone. So this is, as you see, this was published in Cell just on the 4th of December, and in 30 minutes, basically, you can detect this viral with confidence. Last but not least, let's see whether we can use CRISPR not only for detecting, but also as an antiviral. So we need to be aware that there is yet another Cas protein, this time the Cas13D, and the Cas13D is also an RNase. It cuts RNA. But in contrast to Cas13A, Cas13D, it remains specific. It only cuts the RNA molecules that are guided by the small RNA molecules. It doesn't have unspecific activity. With such a capacity, it was obvious, and as early as in March, it was this publication as a theoretical approach in which you could encapsulate in adeno-associated viruses this Cas13D and several guides against genes of the coronavirus genome, so that you can target the cells in your respiratory cells, and basically you can promote the removal of the viruses. This was first demonstrated in April by this group in Stanford, that they also created a very interesting acronym, PACMAN, for prophylactic antiviral CRISPR in human cells, even though what they used, they first genetically modified human cells in culture, and they inserted the Cas13D as a transgene. So these are transgenic cells. So I think these will not have clinical consequences. These will not be brought to the clinic, but they wanted to demonstrate, this was a proof of concept experiment, that once they have the cells that are producing Cas13D, if they're infected with the SARS-CoV-2 or with the influenza virus, which, by the way, it's another RNA viruses, with the appropriate guides, they will be able to interfere with the replication of the virus and reduce. This is actually what we're doing in the lab. But instead of transgenizing the cells, what we are using is ribonucleoproteins, a combination of Cas13D purified together with the guide, and we're giving these to cells. We are selecting guides against the most conserved parts of the coronavirus genome, and we hope this will be eventually converted. So this is a complementary and multidisciplinary approach. I'm not a virologist at all. I never worked with virus. The only thing I know is a bit of genetics, but this is what is happening during the COVID. It's forcing us to think what we can add to the research for doing new projects, new challenges. So we associated with virologists, such as Dolores Rodriguez here in our center. She's working, by the way, next door. We've been together 24 years. We never collaborated before, but because of the COVID, now we are collaborating together with Miguel Angel Moreno. He's an expert in Cas13, and Almudena, and she's an expert in CRISPR. So the last words is regarding the patents. So you know that there is this ongoing battle for the different patents are associated with CRISPR. Suffice to say that as it currently stands in the States, basically the patents seems to be going on the side of the broad, whereas in the Europe, the patents are lying on the side of the UC Berkeley and on the side of the associated companies Caribou and ERS Genomics, that is Jennifer Doudna and Emmanuelle Charpentier. So this is a very complex issue that is, unfortunately, in my humble opinion, preventing the explosion of new techniques. So you can see how the different combinations of agreements between the institutions holding the patents and the associated companies, and how do they interact and interact with for different applications. And just to illustrate, so just to see what is our European landscape, this is October 2020. There are very few institutions that have already obtained they are legalized for using CRISPR for other things other than academia. For instance, in Europe, it's only the German Helmholtz-Zentrum, and in UK, the MRC. That's it. There's no other institution in Europe that has obtained the license for using CRISPR beyond the academics. And now I'm just finishing just by acknowledging my group and acknowledging the different institutions that are providing some funds. And I hope that it was interesting for you, and I will be ready to answer any questions. Thank you.
Thank you, Lluis. It was so interesting to listen to you. That's the reason we like you so much in ERC ethics sector, is that we can say that you are modeling as a person that science and ethics go together.
Absolutely.
So, before handing over the moderation of questions by the audience to my colleague Nicole from the ethics sector, let me take the liberty to ask you the first question. A recent opinion by the European Food and Safety Authority concludes that in the area of plants, no regulations are needed. The current regulatory framework in the EU is deemed sufficient. But in the other areas and on international level, we heard calls for a complete ban, for example, from UNESCO, or calls for an oversight registry concerning research projects, for example, WHO. And we are awaiting, as you said just some minutes ago, an opinion by the European Group on Ethics on all areas of gene editing. What, in your view, is needed? And where do you think are we heading towards?
Okay, thank you, Philippa. This is an interesting question. The first thing to say is that we have to be more accurate. What the European Commission was asking to the EFSA was not what you just said. Was asking whether the existing legislation can be used to regulate the new methods of mutagenesis. And the question formulated as such has to be answered with a yes. Of course, with generosity and with open mind, we can use the 2001 directive to regulate what is happening with this mutagenesis new methods, such as the genome editing tools, which, by the way, they are not introducing new DNA. They're only changing precisely in different ways. That means that the EFSA also said that we need less risk assessment because we are not introducing. And EFSA was also suggesting that we can also consider some exceptions, which is what it's normally requested now by the scientific community. We know that the standard mutagenesis, in which we don't know what's going on when we are bombarding the plants with radiation or we're exposing the plants to mutagens, with thousands of chromosomal abnormalities everywhere. We don't know what's going on, but we only select the bigger pumpkin or the bigger tomato or the nicer tomato. And that's the one that goes to the supermarkets. And we don't do any risk assessment. We assume that these are safe. Now we're using a tool that is producing the same double strand breaks, it's producing the same precise mutation, but we know where these mutations are occurring. So from the logical point of view, from the scientific grounds, it would seem logical that this would also will be considering for an exception, but this is not happening. And regarding the EGE and regarding the genome editing in general, and such as the application of these genome editing techniques in the germline and etc., well, we also have to apply science. And the science says we cannot control the output. And as you have seen in the mice, for instance, when I do an experiment with mice, I'm obtaining 20 different founders, and I line them up on my table, and I sequence the DNA of each one of them, and I pull one because that's the one that I select, and I discard the other 90. How can I do this with human beings? This is not ethically acceptable. And this is the main difference that we need to be aware that divides the academic and the clinical world. And unless we are advancing in our understanding of these repairing methods, the repairing phase, we shouldn't be considering applying CRISPR for human beings. Well, there are some exceptions, and the exceptions are the ones that have been presented. There are few cases where you can apply CRISPR safely because you do this ex vivo. You do this in cells that are outside the patient, so you can select those that are having the correct modification before returning them to the patient. Plus, the only few cases of in vivo are using organs such as the eye. And the eye is a very privileged organ. The eye is separated from the rest of the body by this hematoencephalic barrier. So nothing can escape from the eye. So the potential lateral damage are limited.
Thank you.
Yes, thank you very much. So we're waiting for questions to pour in via the YouTube. And yes, so you presented all the potential of the technology, some of the controversies, and also the situation we're here in Europe. Could you say a bit more to the oversight mechanisms that are being proposed in the human sector? So I'm taking up the question
Yes.
Philippa already asked.
This is important because I think we all understand that we shouldn't be doing radioactive experiments at home. And we cannot do this because there is a law that prevents this from happening. We need to have an installation, we need to have an equipment, we need to hold a license, but nothing like this is happening with the genome editing. So I invite you, any of you, to just go to any of the hundreds of companies that are distributing CRISPR tools, and from home, you can use your credit card and you can purchase these reagents, and the reagents will be delivered to your place. So we haven't yet realized about the power of these techniques, which can be used for good, but can also misuse, as we just seen. So I think we need to be aware that we don't need new legislation. What we need is to adapt the existing legislation. So this is what we have to do. So for instance, regarding this GMO directive, well, the GMO directive was discussed during the 90s. And this is 15 years before we can even think that CRISPR would appear. So it is logical that nowadays in 2020, we probably have to at least attempt to modify this directive, or at least modify the way we interpret the directive. I'm aware, I'm not naive, I'm aware how complicated it is to modify directives and nowadays with the different member states and etc. But at least we need to try, because otherwise there is no regulations. For instance, what happens to this Chinese colleague that is now in jail was not condemned because he was applying CRISPR to human embryos, as we might have thought. He was condemned because he was acting as a physician without being such. He was acting, so he was interfering with this professional status, and he was pretending to be a physician. And because of that, he could be condemned, because that was the only existing law that could be applied. So this is already a clear demonstration that we need new rules that will govern. And also we need to call on the ethics committees and in the ethics advisory boards that whenever a genome editing experiment is being considered, it needs to be explored, it needs to be assessed. Even when we're doing experiments with mice, and as I just said very briefly in passing, so the risk in doing the genome editing experiments with mice is that we can do hundreds of mice. And then we need to be aware that the three R's still apply. So the reduction still applies. So we need to be very confident, and we seriously consider that we only need to produce the animals that are exactly needed, not any other animal that is not needed. So this requires this ethics oversight that Philippa was also referring.
Thank you. So one question from the audience. So Xavier Saoui is saying that it's a great presentation, asking the question whether you think, but don't you think that off-target effects could introduce unexpected threats when editing plant elements cells, let alone human cells?
Okay, the off-target effects. Much has been written about the off-target effects, but this is not the main problem. Off-target effects are very easy to deal with. In the animal and the plant kingdoms, basically, you just have to segregate them apart and finito, basta, enough, it's gone. So there's no off-target effects anymore. In the human cells, of course, because in the human cells, we don't have the breeding steps. So we need to be sure that we are only targeting the desired gene. So this is why we are advancing into different directions. We're using better and better bioinformatic programs for designing guide RNAs, and we also using new specific nucleases that they are even more specific than the ones that are existing. So definitely off targets, this is not something that we have. We need to be aware, we need to care, but this is nothing to be absolutely worried. What we need to be worried is about the on-target effects.
Okay, that was a very clear answer. So another question we have is from Tatiana Oviedo, who works in our ethics sector, asking about the major obstacles to the advancement of gene editing, both in research and the market.
Well, the major obstacles in the research are basically that we're just running out of time, and we want everything to be done today and not tomorrow. And we just, from time to time, you need to do a bit of a perspective and realize that just 7 years ago, we just didn't know anything about this. So in 7 years, we just gone a long way through, and we even have a Nobel Award that has been awarded to Jennifer Doudna and Emmanuelle Charpentier. So this is an amazing speed according to normal research and normal biology. So time, it's the obstacle, because basically there's no limitation. So it's always the imagination of the researchers. Well, the market, we need to be aware, and this is not our responsibility. My responsibility is to assess the politicians and to provide them with the most accurate scientific record, the most accurate scientific information, so that they can adopt the decisions that they wish to adopt, because they have been elected. I've not been elected. So they are the ones taking the decision, but they need to take these decisions informed. It was unfortunate in the 80s, Europe was where the transgenic plants techniques was actually born in Europe. Was actually born in Belgium, by the way, and Europe has never benefited of this technique, whereas the rest of the countries have benefited, and we are purchasing millions of tons of transgenic plants, because we need them, but we cannot grow them. So for the CRISPR, for the genome editing, we shouldn't be committing the same mistake. So CRISPR was also born in the south of Europe, but the battle of the patents, it involves the east and the west coast in the US. It doesn't involve, unfortunately, Europe, even though it's a part of the patents that have been just transferred to Europe. I think we need to transfer this correct information to tell that this is yet another way of improving the characters and the capacities of our plants and of our animals, that we can do things much better, and that we should not be prohibiting and blocking and putting hurdles to our biotech companies. So our biotech companies otherwise will just go somewhere else.
Yes, I'm sure there are lots of lessons to be learned from the GMO discussions. And you also pointed out that it's a principal difficulty to anticipate future development in science for the legislator and in the ethical discussion. We have in Europe, of course, the advantage of also having a very elaborate discussion on ethics and taking in public opinion on that. I'm sure there will be a lot of more discussion on CRISPR-Cas, and as you said, which will be informed by scientists and public opinion alike. So my personal question to you is, before I then go over and read out the other ones, would be on how to best actually to proceed in organizing this public debate.
Well, there's different ways that you can proceed. There is no such an international law. The thing I learned from the lawyers is that The only thing that we know are national laws. So we need to go country by country, but this is something that will take years. So the opportunities are to use agencies that have an oversight over many different countries. United Nations is what we have picked at a reach. So approaching the United Nations and pretending or suggesting that we can modify the declaration of the human DNA genome, for instance, and adding a couple of sentences that will be addressing these genome editing issues might be a way to go. The other ways to go that have been proposed are such you can impose a moratorium. But a moratorium normally doesn't have any great effect, because sooner or later you will have a country that will be no longer following this moratorium. So the question will be, who will be the first to escape from this moratorium? I think with the progress, I think we need to adopt the legislation and adapt the legislation, but we need to adapt it according to our knowledge on the scientific
I get
grounds.
your point.
And this is the only way to proceed. And besides, things are legal or illegal. So if we put the legality of the things aside, so there is always the scientific knowledge. And at the moment, for instance, it would be very unwise to apply CRISPR on human embryos.
Of course. Yes, and so in your talk, you also mentioned the difference between enhancement and the therapeutic use, which in itself is also a distinction which differs from society to society. What about that?
Well, we have different moralities. There is no such one morality. So there are different cultures with different morals, and these different morals, they are informing different laws. So the legislation in different countries are diverse, and we have to deal with this. But from my point of view, from my humble opinion, so working as I work with the rare disease, there's so many millions of people that are suffering from rare diseases that at the moment they don't have any cure, that I think it is obscene to be wasting resources on using these techniques for enhancement, instead of using all our available time and all our available resources to develop potential therapies. I'm not meaning that we might be succeeding in all the cases, but at least we have to try. So, and this is my message in all these different cases. I understand that there are people willing to use these tools for improve themselves, and very naively things that say, I can select the eyes of my kid. I can select the color of the hair of my kid. Well, let me tell you, I just happen to be working for more than 30 years in pigmentation, and I know that there are more than 650 genes that are required to detect or to define a pigmentation. So now tell me whether we can fix these 650 genes at once in order to be sure that our kid will be born with blue eyes. So this is unrealistic. And so this is something we need to be clearly, because this is a regular example that is used again and again in the press. So having a kid with blue eyes. Well, if such a thing would be as easy to do, but it is not feasible to do at the moment.
Right, but that's about the feasibility, and the other argument was about the best use of resources. That this should be, others should be used first. I think that in other places there are also people saying, well, it should be a strict division, and we should restrict it to therapeutic use only, as whatever that is defined in detail.
And also the other thing, Nicole, just if you allow me to say, for instance, when the transhumanists, the most extreme transhumanists, they're saying, so let's kill the CCR5 gene, because in that way we will interfere with the entrance of the HIV. Well, let's not be naive. The CCR5 is not there for the virus to enter. The CCR5 is there because it's doing a very important role for the immune system. It's the virus that has evolved to use the door to enter the cells. So we will be committing a mistake if we can take for granted that we can remove easily the CCR5 function, because CCR5 is required for a proper immune function. Actually, we know that people with CCR5 mutations, they're more sensible, they're more sensitive to influenza and to West Nile virus and to many other infections. So we should be careful, because some of these very easy to transmit, these take-home messages that are transmitted by the biohackers or the transhumanists are just simply not true.
Right. So there's the need also to by scientists like you to communicate on what actually can be done and what cannot be done. What would be your idea on actually dealing with biohackers in general?
Biohackers, this is the question is, what type of resources are you using to do this experiment? You're using your pocket money, your private money, then there's nothing we can do. You are interfering, affecting with the health of third parties beyond yourself. Now the legislation comes into place. So we cannot do anything if someone wants to commit suicide. Well, I mean, we feel sorry, but there's not much we can do. We can inform them that they should not be doing this, but if they want to do this, well, it's at their own risk. But whenever they are crossing the public barrier, for instance, someone is doing something that will affect the community of people surrounding him or her, then we can apply the legislation.
Okay. So we're coming to a close of this fascinating discussion slowly, and so I'm handing over back to Philippa.
Thank you very much, Lluis, for this very, very informative talk. Thank you also to our audience for all the relevant questions asked, as well as to the staff behind the scenes. I think I only can tell you now good afternoon and stay safe while we wait for CRISPR-Cas developing and getting so many possible good solutions. Thank you.
Okay. Safe Christmas to everyone.
Thank you.
Thank you.